Hello and welcome to our discussion on clinical trials. This is our final study design that we will be looking at together, and specifically clinical trials are the only one we'll be looking at that are in the category of experimental. All of the other study designs that we've looked at were observational. So as we are going through this, I hope to share with you the one specific thing that differentiates between experimental and observational designs. So, as always, in a particular study, we start with a population. Since it's often unwieldy and impossible to measure everyone in the population, we take a sample from that population that we think is representative, and then we measure something for each of the people that are in that particular sample. In terms of what we measure, this is going to look very similar to the cohort study that we saw earlier. First of all, we measure an exposure. And this exposure is measured before the onset of disease, again. Then we allow a certain amount of time to go by, and then we measure the disease, or the outcome, whichever outcome it is that we're interested in. And so it's very important to remember that in clinical trials we have two measurement times. We measure the exposure, allow time to go by, and then measure the disease. How then is this experimental study different than the observational cohort study that we looked at previously? The main difference is the defining feature. The thing that makes clinical trials unique is that the researcher assigns exposure. So this exposure element here, not only do we measure it, but the researcher assigns specific people to have that exposure, or in some cases to not have that exposure. Some people think that randomization is the key feature of clinical trials, and that's not necessarily the case. Some people think that blinding is the key feature of clinical trials, and that's not necessarily the case either. It is this assignment of exposure, and that's where both the benefits and the limitations of a clinical trial come in. So first of all, let's look at some of the benefits. As soon as I assign exposure to a specific group, what that does is it diminishes the influence of individual characteristics that might be related to the exposure and or the disease to be the real thing that we're looking at. And so if I assign somebody to have a particular vaccination, or I assign somebody to eat a certain food, and we'll look at some historical examples of those two coming up here, then the other things about that person, maybe their other health habits, or maybe some genetic or personal family or other kinds of things, won't be the real reason that that particular person ended up with the disease at hand. So in a way you can think of it as diminishing the biases that we might start out with when we are doing some kind of a study. So that can be highly beneficial. So that's the plus side. However, there is a downside to assigning exposure, and this whole assignment of exposure can get tricky if the exposure that you're interested in studying is known in some way already to be harmful. And so typically with clinical trials, the main things that we're studying have to do with primary prevention, secondary prevention, tertiary prevention, because we can assign things that we think are going to benefit that particular patient. Some common things that are studied in primary prevention are vaccines, maybe health education, certain intervention programs that we think are going to be beneficial. Remember, primary prevention means the person does not have disease. So I'm going to put here no disease. Over here they do have disease. In secondary prevention they don't know it yet, they don't have any symptoms, and in tertiary prevention they do know they have it, so symptoms and no symptoms. But both of these, they actually do have the disease. A really typical thing that we do in secondary prevention is screening. So we'll be looking more at screening at another time. Tertiary prevention might be a person has the disease, they also have symptoms, and now we're going to give them a drug, or we're going to have them have surgery. All of these things are intended to help the person either prevent that disease or mitigate the disease or improve their symptomaticness of the disease. And so because the researcher is assigning the exposure to that particular patient, that can cause some problems. That brings into play an interesting word that I like people to be exposed to, specifically that of equipoise. Q-U-I-P-O-S-E. Equipoise means the whole idea that we are not sure of the effect of that exposure. And so we're not sure if that's beneficial or not. And so if we have equipoise, then that means that we are able to assign that to a specific person. There are a whole slew of things that we do not have equipoise about. In other words, we know already that they're harmful, or we know already that they're beneficial. If we know already that they're harmful, then we can't assign that to a specific patient. Let's say we take cigarette smoking. We cannot assign a group of people to smoke cigarettes for the next 10 years and see what it does to them. The other thing is, if we know that it's beneficial, we can't assign a comparison group to not receive that benefit. And so a lot of times our comparison group will be what we call standard of care for that particular reason, because we need to make sure that whatever it is that we're doing is giving them the most benefit that we know of that's possible. So if we know it's harmful or we know that it's beneficial, then we do not have equipoise. We already have knowledge about that particular thing. So that is a key feature with this whole concept of assigning the exposures. We need to have equipoise. So let's think about some of the historical cases that are clinical trials. And going way back to 1537, let me draw these up here. 1537. We had a gentleman named Ambroise. Oh, let me see if I can spell it here. O-I-S-E. Paret. Now, Ambroise Paret was a battlefield surgeon, and there have long been wars, and one of the standard treatments at that time in 1537 was if a person got a battlefield wound, that could literally be fatal, because we didn't have access to ways of treating wounds that we do now. People often would die of infection if they didn't die of the wound itself. And so somewhere along the line somebody got this bright idea that if we pour boiling oil onto the wound, then we can help that person to survive. And I don't know how effective it was. They never probably compared that to other things. Maybe they compared it to what they thought was happening at the time. But one night there was a particularly brutal battle, and Ambroise Paré ran out of oil to pour on people after they had these battlefield wounds, and he said, bummer, now I'm going to have to do something. I can't just let them lay there. So he tried something new. He tried a new treatment and then poured that onto the wound instead. And he woke up in the morning, expected all of those people to be dead because boiling oil was the only thing that was going to save them. And as it turns out, this new treatment ended up he went and looked at them, and they were all alive, and they looked great, and they were happier than the people that had boiling oil poured onto their wounds. And so he decided maybe he was going to use this new treatment from then on out instead of boiling oil. So based on what he found with that particular group, he changed his mind about how he wanted to expose people to prevent that bad outcome from happening. Another one that I like pointing out in terms of kind of historical events, in 1747, a lot of people were sailing all over the place in the world. Either they were part of a group that was trading goods between companies, and or they were exploring the world. There was a gentleman named James Lind, who was interested in understanding scurvy. And scurvy is a particular disease that affects people when they are sailors and they are sailing on a boat for long periods of time. They have limited kinds of food that they can take with them, and so a lot of times sailors would get this disease called scurvy, and it would take them completely out of commission, and they wouldn't be able to work, and that would definitely impact the exploration or the trading company. And so if they had these sailors who are sick all the time. So this is one of the first studies that we know of that there was what's called a concurrent control. What James did is he took 12 sailors who had scurvy. So notice that here we're in tertiary, we're trying to prevent something bad from happening. And all 12 sailors were sick. They all had scurvy. And he randomized them to 6 different diets. Just to see, is there anything that we can do for these sailors? We can feed them so that the scurvy goes away. So what they found is that the sailors who received citrus of some kind had the best recovery from their scurvy. So that was a clinical trial, where the researcher assigned this exposure to specific sailors, and they compared the disease among all of the different groups, and those with citrus. After that, what they did is they started sending limes on board the ship because they kept well. They didn't get rotten, and they could eat them occasionally throughout, and they pretty much eliminated scurvy as an issue on these sailing expeditions. And that's where we get the term limey. Sailor is also known as a limey. We have other very notable clinical trials, Jenner with smallpox, a variety of different trials that have been done, and pretty much every drug trial, this goes through a variety of different phases, different surgical procedures or implants or those kinds of things all go through this same idea of a clinical trial. It's worth noting that there is a concept of intent to treat versus treatment received. Now remember here, this exposure was assigned. And this is the key feature of a clinical trial, is that we assign the exposure. The researcher assigns the exposure, and if over time that is in fact the exposure that the patient continues to receive or actually does receive, then that's great. But every once in a while, people receive what's the opposite of what they were assigned to receive. So let's say a person was assigned to receive a placebo, but then they went out and they bought the drug that is the one that's being evaluated, and they took that instead. So they were actually exposed to the opposite, but they were assigned to be exposed to placebo. Let's say maybe we have, in another situation, maybe we have some people who are either assigned to watchful waiting, which means that we just continue to monitor them, versus being assigned to have surgery right away. Well, let's say we assign people to watchful waiting and we assign them to surgery, and one person gets so sick that we realize we really have to do surgery on this person, even though they were in the watchful waiting group. And so they may end up having the opposite exposure as what we were intending based on this assignment. And we do different kinds of analyses then when we go to do this inference about the population. There is an analysis that's known as intent to treat. In other words, we analyze each individual in the group that they were assigned to be versus the actual treatment that was received. And so we might also analyze them based on if they did what we call crossing over into the other exposure. So treatment received is another way that we could analyze that data. So you'll hear these words, intent to treat, and I wanted you to be familiar with that. I hope this has been helpful in your understanding of another study design. Thank you for joining me.